Okay, so our last speaker before lunch, none of us would be here today in this particular room without Dan's help. So we're again very grateful for that and for all that UCLA is doing under Dan's guidance to help with Dup15q. So today he's going to talk to us about functional genomics in Dup15.
Great. Yeah, thanks. It's, I mean, honestly, we're really grateful, as I said before, to be able to have this meeting here. So no thanks needed, both in the Autism Center and in the IDDRC. 15q duplication, thanks to the work of Shefali on the clinical end, Payman on the basic end, is really kind of coming into its own, and our own work that's been kind of chugging ahead, but certainly not as leading as theirs with respect to 15q. So anyway, it's really great to be able to talk to you guys about what we're doing. I'll just go into it. First slide is conflict of interest slide. Nothing I'm talking about is related to this, but I always put this up. How's this? Can you guys hear me better? Is that better? Okay. So, our fundamental so there are a lot of problems we have to deal with in a neurodevelopmental disorder, and certainly something that is as heterogeneous as autism. But this is the case with almost every neurologic and psychiatric disease. There's not that much unique about autism from that perspective. So in some ways, autism has been propelled, when I think about things like Alzheimer's disease that neurologists and Parkinson's thought were diseases, as I started to work on autism, almost now 20 years ago, it became clear to me that actually neurology was kind of fooling itself in a way by thinking of these as unitary diseases. In this, we can learn a lot from the psychiatric disorders. I won't belabor that point too much, other than just to show that there are multiple modes of inheritance of autism. And then again, when we started, and Sarah was in on this pretty much at the table with Cure Autism now, and I think most autism advocates at that point had the same idea. Well, we're not a Fragile X society, we're not a 15q duplication society, we're an autism society. So those things are outside, and it became clear within a couple of years that that was not correct, because some of those kids of the founders of these organizations and stuff end up having these rare disorders. So at some level, we've begun to view autism, at least a part of it, in part due to a collection of rare disorders, and 15q duplication being what, in our experience and agree, the most common form, both de novo and inherited, of these very rare disorders. But, it's not the whole story, and in that if we look at that pileup, and this is a review that was written by Luis de la Torre Ubieda, who was standing in the room somewhere, I don't know, now he's sitting, but it was led by him and others in the lab, but all the great art is due to Luis in this. But, putting together, and Jason Stein, putting together this, if you look at the, here, I'll just go over here. Well, I won't. I'll tell you what, I'll try to point on this. This is kind of the whole risks profile of autism, and you can see that these rare conditions make up, that we've identified so far, make up about 3 to 5%, depending on how you look at it. There are different ways to think about it in terms of population risk versus percentage of people with it, because it's pretty clear that, again, some of these have very high impact, but they may not be sufficient on their own to cause autism. Why do only 20% of Fragile X kids, roughly, somewhere in that range, actually come out with an autism spectrum disorder? That's still incredibly high penetrance for autism, but there are all these interesting questions, and the key point here is that most of the genetic risk so far is undefined. But we can say that at least 50% is going to be of autism as a whole is going to be due to common genetic variation. We can estimate that even if those variants haven't been identified. And I think one of the critical questions going forward from a genetic standpoint is how much does polygenic background, or these a mixture of low effect size common variants work with these rare variants to result in individual phenotypes. For 15q, I don't think it's as big an issue actually as it is for the other conditions, because in my, just reading of the literature, and again, raise your hand and say, nope, you're wrong, is that this has extremely high penetrance. It's one of the higher penetrance conditions for autism, or what we call autism. And again, autism is incredibly heterogeneous, and as Shefali and her lab and other collaborators and others are pointing out, that there are subtle differences, potentially, or subtle distinctions between a 15q form of autism and maybe some others, but that doesn't mean it's not autism, because autism is such a broad and heterogeneous. So genetic testing can identify about 20% of mutations contributing to autism. And again, you can argue whether it's 15% or 30%, but it's somewhere approximately 20%. And so genetic testing is clearly indicated in any case of autism, because again, we've seen these huge variety in phenotypes. So here we are, actually it's kind of amazing. There are dozens, if not hundreds, of autism candidate risk genes identified with some statistical evidence to support them. If you use an FDR of 0.5, we're talking over 400. If we're talking about an FDR of 0.1, we're probably close to 80 now. So, of a large effect size, rare variants. But yet, so this is in a way, one has a basket that's pretty full or getting full. We still have some more genetics to define as shown here. But there are huge challenges to the genetics paradigm. There's extreme heterogeneity. Genes don't act in isolation. They're components of signaling pathways, and in the brain, complex neural networks. So can we identify areas of convergence within these large dimensional data sets that are related to key aspects of function or disease pathophysiology? And can we use these patterns that we might identify to inform mechanisms of treatment? So again, this has been a long-standing view in our laboratory to not study one gene at a time, but to try to understand convergence. And the genetics really necessitates that we do that. So not that don't get me wrong, it's not that the study of individual mutations isn't important, it's actually critically important, but the piece that we've been focusing on is kind of how they relate to each other in a way to try to find convergence. So again, we're not here yet at this level of integrating all of these levels from kind of sequence data all the way up through clinical phenotypes together, but we're actually moving in the right direction. I think there's substantial progress here. In addition to these kind of issues, and I just want to highlight this notion of, this genes don't act in isolation, they're components of biological signaling pathways and in the brain neural networks. This is really framing the problem. This is the problem that we're all working on together in a way. You have genetic variation, and in the end you have a phenotype. And the interesting thing is that the phenotype that we're talking about is a clinical diagnosis that involves behavior and cognition. So we're even kind of beyond behavior and cognition when we're talking about clinical disorders. And one of the critical things is to identify those dimensions of behavior and cognition that are critical, like motor function, et cetera. But what we have to do is kind of go from DNA to RNA to protein. Of course, between DNA and RNA, there are all the regulatory epigenetic things that occur, to kind of cells and circuits and synapses, et cetera. And can we do that type of connection, and how do we do that? One of the things, and I won't go into it in gory detail, I'm happy to show you evidence of it, is that what's kind of interesting is that everything that we're talking about, every level here, actually can be described mathematically in a graph theoretical framework as a kind of small world network. They form scale-free networks, and therefore, even in a unit kind of independent way of looking at this, putting these together is really plausible using kind of network biology and thinking about it in that way. And what I'm going to talk about today mostly is our work on mRNA as a kind of part of the molecular hierarchy from gene all the way through to neurobiology. The RNA being let's call it an intermediate phenotype that's quantitative and measurable and high throughput. So genes don't act independently, they form networks. So again, we don't have to look at every RNA molecule independently. If we look, there's a correlation structure to the data. They're co-expressed. From the correlation, we infer co-regulation. We can identify groupings or clusters or modules of co-expressed genes, and it turns out that these genes that are co-expressed correspond to elements of biological function. The example shown here from a review that we wrote almost 10 years ago now is if you take cortex, and this was published data, microarray data, from 100 different people or 200 that other people had published, and you do gene expression profiling, you can identify co-expression patterns that reflect the underlying biology. And that biology is again constructed by understanding the sources of variation. The main source of variation in the tissue is cell type. So you can imagine that between individuals, the actual percentage of neurons, either because of the way we dissected it or because of biology, doesn't actually matter, ends up, this correlation structure ends up pulling out those main sources of variability. So we identify genes that are expressed in neurons, different neuronal subclasses, different glial subclasses, endothelial cells, et cetera. And so you can do an in silico dissection of a hunk of tissue in this way. And so you begin to get even cell type specific inference. Just one example. Does that make sense? Does anybody want more clarification on that point? Because it's really a critical point that, now, if I designed the experiment differently and just pulled out a specific neuron, of course I would not be getting modules that I get out of tissue. The biological problem there is different. But then the question is, what's the major source of variability between neurons? Stuff like firing rate, synaptic density, dendritic length, those things. And those are the modules that we pull out if we do cell types, et cetera. So just to give a little bit of more flavor about that. So any kind of neurogenetics paradigm that's trying to connect genes to behavior in the end must involve connecting these hierarchies. This is the challenge we have to deal with, if that's what you want to do. So we can't ignore this. We can't just study our individual thing in isolation. We have to be thinking about connecting these, and if we're not actually working on this problem. We're working on something else. That will be, I say that really strongly. It's not a belief. I think it's actually just a fact. If we don't, we have to figure out how to do this. This is our job as neurobiologists now who are working on disease. This is an incredible challenge. But we can't just turn our heads and ignore it just because it is incredible. And because we don't understand how the brain works and how these circuits form and everything, we actually need to do that. And that's why, it's just so critical. So studying the mRNA, we've gone to then, so we asked the question, again, about a decade ago, is there a molecular pathology that we can define in autism using gene expression profiling using microarrays? And this was the first paper. Irina Voanagu led this. It was a really brave piece of work because my assessment at the start was autism is very heterogeneous. Therefore, especially in the tissue of where this is going on, we're not going to identify a shared pattern. We're going to identify subgroupings, maybe 6 or 10. And with 20 samples, we're never going to have power to find anything. Instead, what we saw, and once we saw this, I immediately knew it was really important, but it took us years to kind of really begin to understand it and to replicate it, is that with this small number of samples and 2 cerebral cortical regions, there was a coherent differential pattern of expression. And what's shown there is a clustering. This is clustering by genes that are differentially expressed. The genes are here, the patients are here, and the controls. And the controls are in I think here in this particular one they're black and the autism is red. And what we see is an upregulated group of genes and a downregulated group of genes. And of course it's about 400 and something genes depending on where you put the threshold. And so one of the questions here is how do you begin to make sense of groups of 400 or 1,000 genes? And of course I'm going to get back to this network stuff to show how we do that. But even just looking at the up and down, we could see that the downregulated genes were heavily in synaptic genes, things that were involved in vesicle recycling, axon outgrowth, and synapse signaling. And then genes involved in inflammatory pathways, specifically those involved in immune regulation and in microglia and astrocytes. So we went on then to do a network analysis so that we could maybe understand this a little better. And hopefully it will make sense in a moment. So I was mentioning this term scale-free topology, and I just want to explain it for a second because it might, I think when you explain it in a more intuitive way, it makes sense. So look at these two maps. So one is an airline map and one is a road map. And you can see inherently in these two maps that are of essentially the same, the US, different parts of it, that you can make different, they contain different kinds of functional information. They're fundamentally different networks. And the graphs above are essentially cartoon descriptions of these networks. So the airline network obeys an inverse power law. That is, the frequency of connections, K, is inversely related to the number of connections. What that means is that you have a few nodes with very high numbers of connections. That's like a hub, like an airline hub. And that's shown there. And just the inference there is, and I had this terrible experience 2 weeks ago in DC, where, if one airport has a fire in their control tower, it literally shuts down all the DC airports and Chicago, all Midwest, and traffic on the eastern corridor. So these hubs are very sensitive to attack. They are the kind of core. And so if we can identify within groups of genes that are differentially expressed these hubs, they may be kind of the core elements that we really need to be attacking. So scale-free networks show a small number of nodes that have a large number of connections. Most nodes have very few connections, and the few highly connected nodes are essential for the integrity of the network. And if I remove Birmingham, if I, like with a neutron bomb or some, whatever, ISIS, whatever, you can go around it. You know what I mean? It doesn't destroy the whole network. So fundamentally different kinds of information, and the fact that biological systems obey this kind of this logic and these relationships actually allows us to to do this. So in Irina's analysis, she did an applied unsupervised network analysis where you basically ask, I want to identify the genes that are co-expressed. So here in our network, rather than each edge between two nodes being an airplane flight or some kind of route, the edge is a statistical relationship of correlation or co-expression. How much do they go up and down together? So you take that data, you build the modules, you build the network, and then you ask, what am I looking at? So you go from thousands of genes to about 20 modules, which actually reduces the problem, a lot of problems to, which I won't go into, but again, it's really nice to not be looking at 20,000 independent entities, but now only have 20 things that you're testing. So these are some of the covariates shown there: age, sex, PMI, region. And there are these M12 and M16 modules that were both up and down. I'll show you here. 2 modules associated with autism. The one that's down is the M12, and the one that's up is the M16. It's not up in everybody, it's not down in everybody. We see about 2/3, in this paper, about 70% of the patients showing this pattern. This is the M12, and it was enriched in a lot of neuronal signaling genes, CNT and AP1 actually, but FOX1, RBFOX1 or A2BP1 was one of the main hubs. It's a splicing factor. So we validated this regulatory hub by basically looking in those, doing RNA sequencing on those that have low and high A2BP1 or RBFOX1, and showing that indeed the splicing was affected specifically in relation to the level of RBFOX1, indicating a likely causal relationship. The other thing we looked at is, as I mentioned, I'm not sure I emphasize this enough, but gene expression in the brain, like any neuropathology, let's say amyloid deposition, is just a phenotype. You don't a priori know if it's causal or not. It's just related to the disease. So we can't say that this is causal. We can just say this represents what happens in a postmortem brain of somebody who has autism. And certainly, we don't even know if it's an early phenomena or a late phenomena, because in this study, we really couldn't look at age trajectory. We saw no relationship with age, but we did not have very many young samples. So we could just say you have genetic mutations, you have environment, whatever it is that causes the autism. We actually had one patient with 15q duplication here who clustered with the idiopathic autism samples, and that was a clue. And the only thing we could say is, you have these mutations, you end up with this pattern. So that was even surprising in and of itself. But what you can do is you can then begin to get a causal anchor by saying, okay, if I have GWAS data or other rare genetic variant hits, do those hit these modules more than would be expected by chance? Are the genes within this pathway or group enriched in those with genetic signal? And indeed, M12 was largely enriched. We did this by permutation, so the gray is the null and the red is the actual data, the enrichment. But the M16 was definitely not enriched. So we have this neuronal synaptic module that's super enriched in genetic risk for autism, and the kind of glial module, immune module that isn't. However, what was another interesting observation that we I don't think we put in the final paper because we didn't have room, but if you look at the correlation between M12 and M16, it's very high. And in fact, it's higher in the autism than controls. The correlation was almost 0.7 in the autism and 0.2 in the controls. So what we're saying is that these are two biological phenomena that are interrelated. They're not occurring separately, but you have this genetic risk that kind of sits in the neuronal genes, but that the glial stuff happens kind of at very, very close apposition. It's hard to separate them, but the causal anchor would be in the neuronal. That's the model. So our kind of data-driven model is as follows. You have altered neuronal gene expression and splicing, and increased microglia astrocyte gene expression. The neuronal gene expression leads to synaptic dysfunction, which is intimately tied to microglia/inflammation. And this is likely kind of a what I would call a snowball effect, or the microglia inflammation likely magnifies synaptic dysfunction over time. And this leads to autism, but the alternative splicing and the genetic variation lies not in the glial side of it, but on the neuronal side of it. And so that's kind of a model, right? Beginning to try to put causality into this. This can be tested if we have mouse or in vitro models. So the advantage of this approach is that it was totally unbiased. We didn't have a preconceived notion. It's also genome-wide in terms of being unbiased. It's tissue relevant. We looked in cortex, 2 cortical regions, and cerebellum. We saw no significant changes in cerebellum. It's also, however, the weaknesses are it was mostly adult brain, it was a small sample, and it wasn't RNA sequencing, it was an incomplete transcriptome. So we replicated this recently, and again, this was another enormous 5-year task that was led by Neil Parikshak, but also involved substantial contribution from Vivek Swarup and Grant Bellegarde. Neil actually went to the autism tissue program about a month before the freezer broke. I don't know if this was just lucky, maybe 2 months, and actually participated with Steve Ginsburg, who was running the dissections, in kind of doing the dissections and getting the tissue. In total, we had 113 cases and controls, 3 conditions with up to 3 regions, BA9, BA41, 42, or 22, in other words, temporal lobe, frontal lobe, and cerebellum. And we had 9 patients who had a diagnosis of 15q duplication. This is, we did the analysis separately for the different tissues. We had separate matching controls for autism and duplication 15q, so we could kind of use them as independent samples. We had some young samples as well that enabled us to really see if this signal replicated, which it did. So it allows us to compare between regions, between idiopathic autism and 15q dup. And for those who are aficionados, I just want to say one thing. We've worked for years on RNA sequencing analysis, and any of you that are getting into it, you're going to mess up. You will, unless you have people who are really, really expert and have thought about this. Either not just a statistician, but you need a statistician who's actually been involved in this, because this, different than microarrays, there are an enormous number of confounding factors that have to be controlled. And tons of the data that we see in not the data, but the analysis in the public domain is fraught with, we can show is driven by some of these confounders or batch effect and things like this, even in good journals. So be extremely careful. And so we analyze these data in multiple different ways. We use different ways of calling the transcripts. We use different ways of modeling. And, we get a 70 or 80% concordance, across all of these. And that's why it took so long. Grant had to do it his way 2 or 3 times. Neil did it his way 2 or 3 times. And we had to just make sure that it was concordant, that these findings were relatively robust to the methods used, the ways of normalization, the ways of mapping, the ways of calling transcripts, which those of you who do this kind of know it's quite interesting and problematic. But what's interesting is that if you take the first principal component of gene expression, I'm just going to rush through this to get to the 15q because it's published, that in the discovery sample and our new autism sample, those were highly correlated and different from control, and the replication involves quite a few younger samples. So we see this across age. And this is just showing after we've run the linear model. 4 minutes. Unlikely to be finished, so I'll take a few extra. Okay, well, I'll tell you what, I'll skip all of this because this has been published, but diagnosis is what's driving this. So then, yeah. People can leave too. Yeah, but thank you for the timing. So, sorry. So, now we have about 45 samples without 15q, the 915q, the controls all matched. And so we see this robust differential expression pattern in 2/3 of cases. We see splicing changes. We're able to analyze splicing, and this is not in glia, it's in neurons, and that fits with the A2BP1, RBFOX1. It's all these neuronal specific splicing factors. And the pattern is really shared by 15q duplication, which to me was a major, major finding. And I'm happy to discuss why. I hope we explained it in the paper. I'm sure the people in the room understand why as well. But here's a genetic form of autism. We don't need to give these folks an environmental hit or some other gut microbiome or anything like that, right? It may be contributing in them, but they have a genetic hit that causes their disorder. And yet we see all these changes, including the immune inflammatory changes down the road, which is interesting. Another thing we saw is that developmental trajectory was quite interesting. And I'm going to focus you on the M19. This is the microglia module. This is what happens in controls. This is the gene expression principal component first of that. And what you can see is in autism, it just basically, it's upregulated, but it's not, it's an alteration in the normal developmental downregulation of microglia. In other words, astrocytes and microglia stay persistently upregulated when they shouldn't be. So there's probably abnormal synaptic plasticity and kind of eating of synapses that's going on for years. But that's maybe good in a way because this tells us we have a window here where we might be able to affect this, and this could be beneficial. So how do these changes relate to 15q? So these are the patients. It's in the paper. I'm just going to skip through it. Most are isodicentric, as far as I can tell. This is an independent. There's only one of these samples that was in the Buonagu et al. Now, we take temporal lobe, frontal lobe, and cerebellum, but not all of these cases had all the tissue. Well, they had the tissue. It just, if the RNA wasn't good, we didn't use it. So we don't have all of the samples from all of them, et cetera. But that's the number of cases. And this is in a supplemental table. You can go take a look at that. And this was just shown, so I'll go over it very, very quickly. It's super interesting to me. No ASD genes, so like when we look in the autism idiopathic samples, they don't have dysregulation in this region. But you look at 15q, this is a pretty interesting map of what's upregulated. You can see the GABA cluster, the HERC2, HERC-C2, but even some that are outside or kind of like really distal, like SYFIT1. We've even seen some patients who have an interstitial duplication that doesn't involve it. It's still upregulated. So like there are a lot of candidate genes for the phenotypes in this region, which makes it incredibly complicated, but makes me think that we need to take the duplication, have the whole duplication, and then just take out genes to see which are necessary. Wouldn't tell you sufficiency, but anyway. So that's a map you can look at and use. Most genes are upregulated, but these SNRP-N, that whole SNRP complex, the small RNAs isn't. But it's not just differential expression that's shared; it's the splicing. And The other point, this is the splicing on the bottom, that's differential expression. You can see the slope is very high. So these changes are stronger or more robust. They're larger, and in the 15q. And the same thing with the long non-coding RNA. We see the same pattern. So, and another very interesting story that's part of this is that there are 20 primate-specific long non-coding RNAs that are upregulated. And again, just to show this, if we take the first principal component of differential gene expression, you can see that duplication 15q is the strongest. ADI are confirmed autism then, ASD supported by records, but where we don't have an ADI that was done post-mortem, a little bit less. So the more you're kind of certain about the diagnosis, the better the signal looks. So that's essentially, and then we can talk about what we think that means in a moment. But I can tell you that the splicing changes overlap strongly with changes in RBFOX1, NOVA1, and SRRM4 changes that are activity dependent. So some of this we think is a reflection of strong and consistent alterations in neuronal activity that is leading to these changes. Another thing that we've done in the lab, and I'll end with this, is create and characterize a SYFIP1 overexpression mouse. This is a long and torturous story. It started with Brett Abrams, who was in the lab, and Eddie Herman. They made this mouse, and then Asami Ogoro actually characterized it, and then we realized halfway through that we had lost the mouse. That genetically, it was a back transgenic. It was all the copies were there, but the locus was likely had been silenced. So we remade it with that in mind, and that took another 4 or 5 years. And this is work that was started by Jamie Berg, and now is last year Catherine Kugler has done or Katie as we call her, or she has us call her, has done enormous amount of behavior, huge N's, and we have these 2 lines that have similar copy number. They have increase at the RNA level and at the protein level. RNA more so than the protein, but the protein is about 2-fold at SYFIP1. Here's a summary. This is an unfair graph to show because the ends here are very large because we wanted to be sure. But this SYFIP1 overexpression in a mouse on the genetic background that we're looking at, which is C57, black 6, it does not affect most of these phenotypes in a substantial way, other than something that we see changes in ultrasonic vocalizations, but only at very, very early. Once you get to 6 to 9 days, we're not seeing it. So let's just put an asterisk there. But very, very strong and reproducible changes in fear conditioning. Classical fear conditioning. And happy to, Katie can describe that more and may have shown it in a poster last year at the meeting. So we've also done gene expression, which is so SYFIP overexpression doesn't cause a mouse to look like they're autistic. It causes a fear conditioning phenotype. Now, it's interesting. We don't think it's anxiety per se because in open field and elevated plus maze, they don't show pure anxiety phenotypes. But some of it's something about fear and anxiety because once they've learned, once they get the tone, you can just, they look to me, like the data looks, Katie can discuss this more, but there's a anxiety phenotype. And again, it doesn't cause them to have problems in the elevated plus maze, and so pure anxiety, but there's some kind of maybe learned anxiety. So we've done gene expression now, RNA sequencing, 4 animals, genotypes per sex per line, so it's 8, balanced for sex, 64 animals, cerebral cortex, very few genes differentially expressed at P21 or P60. And you can see there's no overlap at P21 or P60 if we use a stringent differential expression. Then if we ask what are these genes, it's hard for you to see, but the ones that are up at 21 shown here, and the down ones are neuronal genes, and the up are kind of astrocyte oligos. And the ones at 60 days, there is a small microglial signature. Some of the interesting genes are here. This is work that was done by Aaron Gordon in collaboration with Katie. This gene we recently identified as CNV in autism that we published in American Journal of Human Genetics last year that is strongly associated with autism and language phenotypes, deletion of this gene. And this is a gene that it's a long story, I won't go into it. But it was of interest from 2005 on. And this is the first association with disease. We reasoned that genes that were expressed in language cortex in humans would be important for language phenotypes. So the first one we worked on was CNTNAP2. The second one, which we never worked on, is NR4A2. But recently we've been able to, it's a coincidence. So now I have to end. So what does this SYFIP1 mouse look like? Well, this yellow module is the upregulated module associated with disease, and it's the only one that's really connected to any of the changes that we see in autism brain. So again, this is just, there's interesting biology here, but the point is we're not recapitulating 15q duplication, we're not recapitulating autism. So in conclusion, autism is heterogeneous, but we can find molecular pathology. Now the critical thing is to connect. We have to go, all the way from, this is a summary of that data. So we have all these findings. How do we connect them? How do we go from the genes to here? What is the pathophysiology and how can we intervene, right? And so one of the main thoughts is using these networks to inform biology. And again, we've developed a framework to ask how do in vitro models model in vivo brain development, and this is work done by Jason Stein and Luis de la Torre-Bieda, and I just refer you to the paper, but I'm happy to talk about it. We've developed a set of tools that allow you to take an in vitro model system and ask, what stage is this and how is it modeling? Okay, and so we can do that now. And working with Sergio Pasca at Stanford, he's developed a series of three-dimensional cultures that actually look more mature than the two-dimensional cultures or the typical organoids. And so I think, and again, this is a kind of framework to be thinking about, but if, from a patient organization standpoint and from like if you wanted to really move this forward, what one has to do is go from these penetrant risk loci like 15q all the way through the cellular, molecular, structural, physiologic changes, identify the convergence in vitro in IPS-derived three-dimensional models, which I think is plausible. You can also layer on top of that polygenic risk if you want to. But the other thing is to really be cross-comparing the in vitro models back and forth between in vivo ephys and behavior and see if you can make those connections. See if one can find a particular spine phenotype, cellular phenotype, physiologic phenotype in vitro that actually matches that in vivo, and then you're off to the races. Of course, this is much easier said than done. But anyway, this is kind of where we're trying to go with this in collaboration with a large group. This is not a one lab thing. This is a 4 to 5 lab thing minimum. So this is what we're trying to do. And so I'll add now, I mentioned most of these people as we went along. This is an older picture. I apologize for that, but I mentioned the work by Katie and Aaron on the SYFIP1, the work that Neil, Vivek, and Grant really led on the co-expression. I briefly mentioned the stem cell work that Luis and Jason have been doing. And then our other collaborators, Ben Blanco, who helped us with splicing, Sham Prabhakar with epigenetics, and Jonathan Mill continues to do methylation studies on these brains. So thanks for your attention.